Hello friends, this video on applications of biotechnology part 7 is brought to you by examfear.com. No more fear from exam. Let us try to understand how genetically engineered insulin was produced, how the process exactly takes place. So, with the help of genetically engineered insulin, these advantages which were associated with the insulin extracted from animals were overcome to a large extent. For example, now the process was not that complicated, it was a simple process because growing bacteria on a large scale was not really difficult. Again, it is actually a simple process and easy to grow large quantity of bacteria and also make insulin is as much quantity as we want. Also, since human insulin was getting prepared, so there was no risk of allergies because it is the human insulin. So same thing is getting injected into the human body. So the chances are very less that the body will react in a different way. So there are no risk of allergic reactions. So now, before we talk about how insulin was engineered genetically, first let us look at the structure of insulin, how the insulin looks like structure-wise. Now, it has two short chains of polypeptide. Now, what is insulin? Insulin is a hormone. And what are hormones? Hormones are nothing but proteins. So, insulin is also made up of peptide chains. So, there are two peptide chains, that is chain A and chain B. And they are relatively small peptide chains. Now, in fact, they are polypeptide chains, lot of peptide units joined together. And peptide units are made up of the amino acids. So, the structure consists of these two chains, and these two chains are linked together by disulfide bridges. So, here you can see this is chain A, this is chain B, and they are connected together by disulfide bridges. So, this is a linkage between the two. So disulfide bridge, that is there are two sulfur. So the bond between two sulfur, that is why they are called disulfur bridges. Bridges because they connect the two peptide chains. So this in simple words, this is how the structure of insulin look like. Now, insulin is synthesized as proinsulin, which gets processed to form fully mature insulin. Now, initially, insulin is synthesized as proinsulin. Like how we have proenzyme, prohormone, similarly we have proinsulin. So proinsulin is a prohormone. That means this prohormone further needs processing to undergo some change in its chemical structure to form the mature hormone. And the same thing happens here. So if you look, this is the structure of proinsulin. So if you see in proinsulin, you actually have three polypeptide chains. One is chain A, so this is chain A, this is chain B, and here the gray one which you see is chain C. So you actually have three chains and you have the disulfide bridges connecting them. So that is the structure of proinsulin. But when it gets processed and when it matures, what happens is the chain A and B remain together, whereas the chain C separates out from A and B. So A and B remain together, but C separates out from them. And this is the structure of insulin. And this is proinsulin. So the C peptide chain is present in proinsulin, but it is not present in insulin. So this is how insulin is genetically engineered. And I hope you understood the significance of genetically modified insulin. So now we do not have any fear of how will we arrange human insulin, because it is a simple process now. We just need to grow bacteria and get insulin out of it. What is the exact process of genetic engineering to produce insulin? How exactly the bacteria is going to produce insulin? So let us look at the process of production of genetically engineered insulin. Now, first of all, as I said, it is prepared from bacteria. So, first of all, a bacteria cell is taken with plasmid DNA. Now, I have mentioned this before also, that inside a bacterial cell, you have a chromosomal DNA, and independent of the chromosomal DNA, you have circular DNA, which are called the plasmids. And these plasmids have the capacity to replicate independent of the chromosomal DNA. So that is the speciality of the plasmid DNA. So we first take a bacterial cell with plasmid DNA. Now what we do, the plasmid DNA is cut with the restriction enzyme. So if you remember the restriction endonuclease enzyme, this acts as the DNA scissors. So this enzyme will cut the plasmid DNA. Now how is this phase which has been cut utilized? Now this plasmid DNA is this one. Now the human gene which is responsible for insulin production is inserted into the plasmid DNA. Now this is the human gene which is responsible for insulin production. So this one is the human gene for insulin production. So what do? This gene will combine with this plasmid DNA to form the recombinant DNA. So now when this both has combined, it forms recombinant DNA. So this was our genetic engineering or recombinant DNA technology. So now we have this recombinant DNA which has our desired gene. And which is our desired gene? The gene which is responsible for insulin production. That is our gene of interest, or that is the desired gene. So now this plasmid DNA has that particular gene, and also since it is a part of this plasmid DNA, it has the capability to self-replicate. Now what will happen? Now we have our rDNA is formed, that is the recombinant DNA is formed. So this yellow portion is for the gene of interest and the red portion denotes the plasmid DNA. Now what do? This recombinant DNA is introduced into a new bacterium. So there was one bacteria from which you extracted the plasmid DNA. Now you search for some other bacteria, a new bacteria, and you insert this rDNA into that bacterium. So you see this is the bacterial cell. This is the bacterial cell, and you have inserted the recombinant DNA inside this bacterial cell. So that means now the gene of interest is a part of this bacterium. So the bacterium acts as a host, but this gene of interest is also a part of the bacterium. So what will happen? Now as the bacterium multiplies to produce more bacteria, all of them will have this gene of interest. That means all of them will have the gene which is responsible for producing human insulin. As a result, all of these bacteria will produce human insulin, and that human insulin will be utilized for our interest. So that human insulin will be utilized for treating patients or treating human beings who are suffering from diabetes. So this is how exactly the insulin is genetically engineered. So now you understand how recombinant technology can be helpful. So we just need to decide which trait we want or which gene we want. Once we know that, we can just combine it with a plasmid DNA to form recombinant DNA, and that recombinant DNA can then be inserted inside a host cell, and with that host cell it will keep on multiplying, and more bacteria producing human insulin will be formed. And this is how human insulin can be produced on a commercial scale. And it is extremely easy to have a culture of bacteria, because they really don't have too much of terms and conditions for growth. So it is very easy to handle bacteria. So this is how genetically engineered insulin is produced. Thank you. Please visit www.examfear.com to watch more educational videos with a better experience. Please do not forget to like and subscribe to our YouTube channel for latest updates. Thank you once again.